Yes, thank you so much for having me here. It's quite an honor to speak to a room full of people who undoubtedly know more about mRNA technology than I do. But as Dr. Kuritzka said, I wrote this story in 2021 in which I tried to give a comprehensive history of the backdrop, essentially, to the innovation that led to the development of the successful COVID-19 vaccines. And so I think that's kind of the main reason why I'm here. But I've been covering mRNA as a journalist for a while, actually. So going back to 2015, I wrote, I think, one of the kind of the first critical stories of Moderna. Well, not critical, but investigative. And then a couple things before the pandemic, and now obviously since mRNA is the hot thing, a lot of stuff since the pandemic as well. A little disclosure slide, because I know that's what one does at an academic conference. So it's true, I did do a PhD many years ago, but I should say I stand here before you as a humble journalist. I'm not a historian. I'm not a patent attorney, because that's obviously a big issue around ownership. I'm not an active scientist either. But I am, and a little bit more about me, I am now, and I can say this honestly, I'm a movie star. And I have a credit on IMDb that I'm really proud of. And I know movie stars, so here's a picture going way back this is from my high school improv team, which had a couple of famous movie stars who have many more IMDb credits than I do. There's me, clean-shaven, a little more pimples, but as the Entertainment Weekly put it, none of the rest of us are famous, but I am on IMDb now, thankfully, and that's because of this documentary that will be appearing on Swiss TV and be distributed elsewhere. And so I just want to start by playing a brief clip from the documentary. It's not of me, but I think it'll set the stage for the history here.
Getting the vaccines invented, sort of engineered, manufactured at scale, and then putting them through clinical trials, all under a year, and then manufacturing sufficient doses to start distributing them and administering them to patients at the speed at which it was done, is in the top 5 easily of biomedical accomplishments of us as a species. It represents the culmination of 100 years plus of technology innovation in terms of how to build vaccines. It's never been done before at that scale and at that speed.
So I think that's a message most people here know. Again, the movie's The Messenger, coming to a Swiss television set near you. But I want to drill down on the one sentence there about how the development of the COVID vaccines really builds on a century plus, as Dr. Ernab there said. I'm not going to focus on a full 100 years, but I will talk about 60. And I'm going to do it in the framing of this question that I think comes up on everyone's mind every October of, undoubtedly a Nobel Prize will be awarded for the development of mRNA vaccines, and the question is, who do we single out? Which up to three scientists should we pick? And of course, the short answer is hundreds of scientists, thousands contributed to the development of these vaccines. But the Nobel rules dictate 3, and so here are some usual suspects, and we'll get to some of these people as the talk goes along. But I want to talk about one person in particular for the next 5 minutes, if I can, who I think made a really seminal contribution, but is not as well known as maybe some of these other faces, and that's this guy, who some of you may recognize, some of you may not. This is Robert Malone, the original inventor of mRNA vaccine technologies. I'm not the one calling him that. That's what he calls himself, importantly, okay? And the reason you might know him, you might know his papers, but more likely you know him from when he appeared on the Joe Rogan podcast in late 2021, 2022, leading to this huge backlash with Neil Young pulling his tracks from Spotify, because he's one of the main spreaders of what's known as vaccine disinformation. He is an anti-vax, anti-mandate, and as things have gone on since mid-2021, he's increasingly become a darling of kind of conspiracy theory fringe right. But, oh, and here's his Twitter bio. Actually, this was his Twitter bio a while ago when he called himself the inventor of the vaccines. Now it's inventor skeptic. But as he told me back when I spoke to him for several hours kind of before he became infamous, and as he told me, he said, I've been written out of history. So I want to correct the record for Dr. Malone here and go back to the late 80s and talk about his important contributions. So this is a young Dr. Robert Malone as a graduate student at the Salk Institute in the late 80s. And he did a series of experiments, but, oh, there's some formatting problems there. That's okay. Basically what he did is he took synthetic mRNA, formulated it inside a cationic liposome, and delivered it to cells, zebrafish embryos, and mice. So the fundamentals of kind of the vaccine formulated in lipids were there, and he was the first to do that with synthetic mRNA. And also, in this document that he made in early 1988 for, I think, patent disclosure purposes, he says that mRNA could be used as a drug for therapeutic applications. And he's the first person who kind of makes that conceptual leap. Before him, people were using mRNA, either synthetic or natural, and putting it into cells or into xenopus eggs, and just to try to understand development or to probe, try to understand basic biology questions. He made that realization that this had therapeutic or vaccination potential. This is a picture from his lab notebook from 1989. I just think it's cool, and kind of, it's legitimate science there. And he publishes a series of 3 papers. The first one on the left is that experiment with the cationic liposomes to deliver synthetic mRNA into cells. He then does that in xenopus embryos and eventually in mice. He actually ends up leaving his PhD program early for kind of interpersonal reasons, and he goes to work for a small company called Vical for less than a year that he leaves because of interpersonal reasons. And if you want to talk about armchair psychoanalyzing this guy afterwards, we can do that over a beer. I'm going to try to avoid that here. But he gets this patent application, and importantly, buried deep inside this patent that is granted on, you can see there on the right on page 31 and 32, he describes what, very light on data, but it is, I guess, the first example of mRNA as a vaccine. He produces an HIV protein using mRNA, and then he does a challenge experiment in mice to show that it can be protective. So just to recap here, him as the inventor, here are the key things that he did. And this is an impressive list for essentially a PhD dropout to do over the course of 3 years maybe. First, to realize the therapeutic potential of mRNA. First, to deliver synthetic mRNA into a living organism. First, to use cationic liposomes to deliver mRNA. And the first to make these patent claims around vaccination. So here he is on a program that no longer exists anymore, I guess. But just to reevaluate this question, is he the inventor of mRNA vaccine technology? And I've thought about this a lot, and after talking to him for hours, the answer is yes, in a very narrow sense, a very semantic sense, in that in patent law, an inventor is someone who contributes to the claims of a patentable invention. And it's listed there on a patent, inventors, and his name is on that patent, so he is an inventor. But of course, so are his co-inventors on that patent. And in a more holistic sense, in the sense that of science, obviously his work builds on that of others, and everyone had to work, improve upon what he had demonstrated to make the vaccines possible. So coming back to this idea of it takes a village, here's a graphic of all kind of the relevant papers that led to the development of the COVID vaccines, and he's the author on 2, 3 of them, maybe, depending on your database, your index of journals. And of course, his work wouldn't have been possible if we didn't have a way to synthesize mRNA, and that goes back to work from '84 from Doug Melton's lab and others at Harvard. And he needed the cationic liposomes to deliver his mRNA, and that was work from Phil Felgner from 1987 paper there. And of course, that work builds on a paper from the late 70s. This is actually the first demonstration of mRNA in a liposome used to transfect cells. It's natural mRNA taken from rabbits, and it's, a traditional liposome, not the cationic or ionizable ones, but it's liposome encapsulated mRNA put into cells, and that's a paper from the late 70s, which of course builds on earlier work around liposomes to be used as drug delivery vehicles, and liposomes in general, which were discovered by Alec Bangham and originally even called bangosomes. So here's kind of an outline of the history pre-Malone, 1960 to 1990. And I've broken this up into 3 kind of buckets. Stuff that's just about mRNA on the left, things that are about the lipids, delivery system in the middle, and things that combine the 2 on the right. And so those are the faces I just showed you over the last few slides, mapping them onto the timeline. Of course, if we want to go further back, none of this, we wouldn't have mRNA vaccines without mRNA. So we needed to discover mRNA with Sidney Brenner in 1961, and then show that it can be transcribed in vitro, translated. And then here's where Malone fits into the picture. And of course, he worked quite closely with Phil Felgner on those experiments. And here's a fuller timeline of the full 60 years. So 1960 to the pandemic. Malone kind of falls right in the middle, but let's now kind of quickly go over some of the key developments that post-date Malone and that build on his work. On the mRNA side, you had the first demonstration of mRNA as a drug, as a therapeutic in 1992. There might be some problems with that paper. People had trouble replicating it, but it's a demonstration of that potential. You had the first mRNA cancer vaccine in the mid-90s. This is work in mice. And of course, the seminal work of Kati Kariko and Drew Weissman in the mid-2000s, showing that if you modify the nucleotide chemistry, it helps to evade the innate immune system. I'm not going to go into great detail on that, only because I assume the audience here is pretty familiar with their contributions, and so the brevity with which I give to Kati and Drew's contributions is only out of respect to you, and I don't want to take anything away from them. But so I'm not going to go into that now, but that's obviously a pivotal discovery, and we'll come back to that a little bit when we come a later discussion about who deserves the big prizes. I do want to recognize CureVac, because I think they often get overlooked in this history. I think Steve Pascolo is in the room, and he's, there's a picture of some of the founders. He's the young, handsome gentleman in the middle there on my slide. And so they were the first to do a trial of an infectious disease vaccine encoded in mRNA. Onto the right side now, mRNA encoded with lipids. Malone had the first patent disclosures around an HIV vaccine, but Pierre Moulin was the first to publish real data showing, in this case, a flu vaccine, but an infectious disease vaccine encoded with mRNA and lipids. And that was in '92, and then in the early 2010s, we have the ionizable lipid nanoparticles being used to package mRNA, work from Andy Gill at Novartis. Of course, a lot of commercial development that is hard to, it's not always published or it's not as sexy, but so much process development and optimization, and these companies were critical to development of the field, starting with Argos, formerly known as Merix, which was doing ex vivo dendritic cell mRNA vaccines, CureVac, founded in 2000, followed by BioNTech, Moderna, Others, Shire, that became Translate, that became Sanofi. And I think DARPA deserves a lot of credit for funding much of the preparedness work on mRNA vaccines throughout the 2010s that helped enable the pandemic vaccines. The first clinical trial of a lipid nanoparticle formulated mRNA vaccine came in 2015, fully 5 years before the pandemic. That was done at Valera, which was a spin-off of Moderna before it was folded back into Moderna. And then, of course, the emergency authorizations of the vaccines we know, followed by their approval. So that's kind of a whirlwind history of the mRNA side. But I do want to zoom in on the middle column and talk about the delivery system, because I think we call them mRNA vaccines, and so people focus on the mRNA. But in truth, they should be called mRNA LNP vaccines, and you see that obviously sometimes in the literature. But the delivery system is equally important, if not as important, whatever. And so the development of these ionizable lipid nanoparticles comes in the early 2000s and can be traced back to the laboratory. Oh, and as Chemical and Engineering News put it so eloquently, without the lipid system, we wouldn't have the COVID vaccines. And so this traces back to the lab of Peter Cullis at the University of British Columbia. Here's the picture, a cartoon of the lipid nanoparticle system with the 4 ingredients, including the ionizable lipid. As Peter has told me and would say, obviously he worked with a lot of colleagues at a lot of companies, and I don't want to get into the history of that here, but here are a couple of the executives who were key at some of his spin-off companies. And well before the pandemic, we had the first approval of a drug using the lipid nanoparticle system, and that was a RNA interference drug from Alnylam. So that delivery system was key as well. And then there's all sorts of patent fights, and I just throw that up there, 2 ways to visualize all the bickering over who owns what IP, but I'm not going to get into that right now. I'll just sort of acknowledge it. So back to the kind of framing, who deserves the Nobel Prize? Well, judging by kind of the pre-Nobel, the mini-Nobels that have been awarded to date, Katie Carrico is kind of the first person who gets named, and undeniably, I think she's up there. Her demonstration that the modified chemistry can help limit the reactogenicity, improve the tolerability, let you give doses of vaccine that work, was critical, and she's been on the covers of all the magazines, and all deservedly so. And she worked, hand in hand with Drew Weissman. So together, them as a package, they deserve all the credit, and they won the Breakthrough Prize and numerous others. And while it's true, and we might get into this in the meeting, that you can develop an RNA vaccine without the modified chemistry, just as you can deliver these vaccines with things other than lipid nanoparticles. The thing that was ready to go out of the gate and the thing that worked in 2020, in less than a year, used their chemistry and a lipid nanoparticle system. So that's essentially the validated system, and so I think you have to recognize them. Some prizes have awarded it together with Katie and Drew. You see some of the other faces here that have been awarded. This was a Spanish prize, the Princess of Asturias Award. They also recognized Phil Felgner's early work in the 90s on cationic liposomes, the founders there of BioNTech in Ugur Sahin and Özlem Türeci, a founder of Moderna in Derek Rossi, and on the right, separate from mRNA, but Sarah Gilbert, who developed the chimpanzee adenoviral vector system at Oxford. What about Malone? Does Malone deserve to be up there? It's an interesting question, made more challenging because he's sort of made himself completely toxic in the last couple of years, kind of gone anti-establishment. And so this is a graphic from a story about him in The Atlantic that ran in 2021. And they have this great quote, and I couldn't say it better myself, but he's messing up his chances at the Nobel Prize by being so anti-science. And it's true. So he's a hard person to want to fet when he is so outspoken against the vaccines. But I think if you had to pull out the most important innovators in this field. If it was up to me, if I got a phone call from Sweden and they're like, who should we give the Nobel Prize to? And I said, well, if you can expand the Nobel Prize to 4 people, I actually would pick Robert Malone. I think his contributions are that important that he would be my 4th pick. But of course, the Nobel Prize is limited to 3. So my 3, if anyone is asking, has to be these 3. So it's Caddy and Drew on the left for making those key innovations around the modified chemistry, and Peter Cullis for developing the LNP system, because together that was the package that enabled BioNTech, Pfizer, and Moderna to push forward and develop those vaccines in under a year. And so that trio has won a few prizes in Canada and in Asia. And if anyone wants to place a bet, I will put money that that is what Sweden will recognize as well. But enough about the past. We're here to talk about the future, I think. And so I'm excited for me to stop talking and to hear from all of you about the ways in which this technology is being applied for HIV and flu and RSV and cancer and all sorts of ways in the vaccine field. Of course, in therapeutics, mRNA is now being used to encode CARs for CAR T cells, or CRISPR-Cas9 enzymes, or protein replacements, monoclonal antibodies. There's all sorts of interesting therapeutic applications, as well as, the technology we're still in the early days of this technology, and so there's all sorts of ways to improve upon the mRNA sequences through optimization and formulation and dosing and all sorts of things on the mRNA, and of course the delivery platform. The LNPs were basically ported over from a rare disease RNAi drug for delivery to the liver, and then we used it to deliver vaccines into arms. And so there's all sorts of ways that I'm sure we'll hear about to improve the delivery system. And so with that, I will turn things over to you guys, the real scientists, and just say thank you to the workshop organizers for having me, to my editors at Nature and elsewhere for help whipping these stories into shape, the websites that I stole much of the artwork from and without credit, my apologies, and of course my sources. Everything I know is from talking to real scientists and reading your papers and stuff, so thank you. And if anyone's in Boston, happy to grab a coffee, or else there's my email address if anyone wants to get in touch. So thank you.